Okay, so good morning, everyone. What we're gonna do today is first I will introduce you a little bit of what my group is doing and how. And the goal for that is not to show what we're doing, but mostly to show how very current type of research is being done with molecular dynamics. A lot of the things that you have learned so far are very helpful to teach you how to do the simulations and deal with this type of simulation. But if you want to do cutting edge research with MD, you probably need to do more replicas. You probably need to use a lot of analysis tools that are not just a button for a click, so how do you do those things? So today we're gonna show to you. So I introduced you for about 30 minutes. What are the things that we have been doing? You're going to see that we do many replicas. You're going to see that we do a lot of things compared to the experiments. And after that, Marcelo, who's in the back, is going to come and talk to you about how we actually do some of those analyses using Jupyter notebooks and a lot of the stuff that we do in parallel because we have a lot of data to analyze. After the break, we have John Stone from NVIDIA talking to you about why you should use VMD for some of these analysis. We all use MD analysis and Jupyter and VMD kind of combined. There are reasons to use these different tools at different moments. John is going to show to you that if you actually need to do analyses that are heavy, VMD is way faster than any other tool because it can handle these large-scale simulations. So he's going to show to you a little bit how you can script in VMD and how you can do those things. And Diego, who actually just left, is going to show to you how to make the nice. Oh, Diego's back there. He's going to show to you how to make the nice pictures and videos that you have been seeing throughout the week. So how do you take the most out of VMD to produce images? Okay? So let's try then to start with this exascale computing era so that we are what we can do today. So a few years ago, okay, not so few anymore, we started what we call the petascale revolution. So the first machine to sustain petascale was Blue Waters, that was University of Illinois. And that's when we went from simulating things like STMV, 1 million atom systems, 1 million atom systems, to study larger and larger things, all the way to the HIV capsid. That was actually the first big result out of Blue Waters in 2013. So that's a work of John Perilla, who is now at Delaware, who was a postdoc also in the center in Illinois. So you can see we went from with the petascale revolution, went from just simulating 1 to 3 million atoms to simulating this is a 64 million atom system and actually extracting a lot of information from it. So that's back in 2013. So that's just over 10 years ago. A few years ago, that was about 3-4 years ago, I think, the first exascale computer came online. So exascale means it's 1,000 times faster than the petascale, right? So it can do 10 to the 18 float point operations per second. So it can do 10 to 18 maths per second. Okay? So this is pretty impressive, but also if you look at it, it have some differences, from the petascale revolution, the petascale time. The petascale, most of those computers were regular computers that we are used to. Those were the same kind of computer you have in your house, but larger and more of them, very well connected. What started to happen in the last few years is that these computers start to change. We don't have so much anymore of the same type of CPUs. We don't have the same types of GPUs. The GPUs got stronger. We have been talking about that this week, right? NAMD is now GPU resident. Also, if you look, I wrote that here. This supercomputer is based on ARM chips. It's the same type of chips you have in your phone. This is not the same processor that a regular computer from 5-10 years ago. It's the same type of processor from your phone. Okay, so a lot of things start to change. And of course, this is not just giving us a better computer that is different, but it's also giving us a lot of challenges because computing on them is not exactly the same. So the center had to work a lot. So we had to work a lot on making NAMD, VMD, all this software compatible, and also able to take advantage of this hardware. And as you can see, it takes some time, right? Like we're only officially releasing MG3 like last week, but we have been working on it for 3 or 4 years to actually adapt to this new type of machines. And it's the same for Ember, it's the same for Gromax, the same for, so all this software have been working the last 5 years to adapt to this new type of computer. So there's a bunch of challenges there. We have been talking about that, and that's kind of like the message I want to tell you. Like we're working on it, and you're gonna see there's a lot of different things, and you have a lot of things that you might need to learn how to take the most out of these computers. Okay, and we have a lot of documentation on our website. Another thing that is interesting is you might think like, okay, but like I'm not gonna buy a $400 million computer, but also the petascale computing, the cost of that dropped a lot. So a machine like Blue Waters costed, I think, $200 million, the machine. Well, now you're talking about buying a computer that is sustaining petascale for $200,000. So that's a huge difference, right? Like it's 1,000 times cheaper to buy a computer like that after 10, 15 years. Like, and that's the kind of machine that we have here in the lab. So like just in our lab, we have 3 of those. In Illinois, I think we have 5 of those, you know, like, so we use those machines now a lot to be able to do this work that we are doing here. So don't think, my message on that is, don't think that because there's this new revolution of cool supercomputers that is going to have a lot of things, that's not affecting you. It's going to be affecting you very soon. It's like whatever comes in the supercomputer now, it's probably going to be the next generation that you're going to have your desktop in your laptop in a few years. So we need to always adapt to those things. Okay, just to give you an idea, so like for NAMD specifically, one node of these machines is more or less equivalent to 400 of Blue Waters nodes. Nominally, they are actually faster, but those like this nominally here, it's because of a lot of the instructions in the GPUs that are very good for like AI kind of applications. For AI, it's probably way more than 400, like equivalent. But for NAMD, is about 400. Yeah, so what can we do with those things? So like I say, in my group, we are interested in mechanobiology. So you're interested in protein mechanics, how protein behaves under force. And we have been working on what we call in silico single molecule force spectroscopy. And the reason to use that name is because I want to make sure experimentalists and also you guys understand that what I'm trying to do is experiments with the computer. You know, like, so I'm trying to do the same thing that we do in experiments. Okay, so what that means. When we talk about single molecule force spectroscopy, we are talking about things that can be done with atomic force microscope, with centrifugal force microscope, with magnetic tweezers, with optical tweezers, with all those types of equipment, like experimentally. Particularly, I have been working for many years with Hermann Gaub, who was one of, not the father of AFM, but the father of using AFM to pull on proteins. So he was the one who created that. And so the way we do that experimentally, you have an AFM cantilever, and you're going to functionalize the tip of that AFM. That's very, very tiny. You're going to functionalize that tip. So you're going to add a molecule that you have interested in. You're going to have another molecule here that is going to work as a fingerprint to make sure that you're doing single molecule. And then you're going to have the fingerprint, molecule 1, and then you're going to have a glass surface with another molecule that interacts with this one. So you're going to have your molecule number 2 and your fingerprint number 2. So remember, the fingerprints are there because you don't ever functionalize the tip with only one molecule. So typically the tip is like tiny, but not that tiny. So you're going to have a few of them. So every time you go up and down with the AFM, you can grab, make a connection, like make a bond, and pull back up and understand how the bond behaves. But then the fingerprints make sure that you only have one. So fingerprints are molecules that you know how they behave under force. And then you know that, oh, that's the signal for the molecule, so I know it's only one. Because if there are 2, the signal is going to be different. But then you know that you're actually pulling one molecule, so you're doing single molecule. Okay, so at the end, this is the part that you want to study. You study the part in the middle, the rest is all part of the setup, right? What you get from this experiment is a force extension curve. So you're going to get, when the AFM goes up, you're going to calculate the force. And we talked about SMD and those things here before, so you know that what kind of disability you're doing. You're going to go back up, you're going to feel the force of the system. At some point, you're going to rupture that bond, or you're going to unfold the protein, and then that force is going to drop very fast. And then you can calculate, you can measure what is the force and the extension, how much we have to extend to get to that force. Simple, right? We can do that in the computer. So we can do molecular dynamics, and we can do exactly the same thing. We're gonna have our molecule 1, our molecule 2, and you're gonna use a harmonic potential to pull on the protein. So you're gonna use Hooke's law, and you're gonna pull on that system and get the same thing. So this was pioneered by Klaus and also by Grubmiller, kind of at the same time. The papers were submitted about the same time and published about the same time in the 90s. That was about the same time that the AFMs or those things started to be invented, so together with the experiments. And at the end, you get a forced extinction curve, the same way. But there is an advantage you guys are seeing here this week. If you go do the experiment, you get a forced extinction curve, and that's it. If you do the simulation, you get a forced extinction curve, but you also know the position of every atom at every time that you're doing. You know everything else about the system. So if we can get a forced extinction curve that is similar to the experimental one, we can go to the simulation and look, oh, this is what is happening. That's why you see all these 10 peaks before you see the main peak. Because, it's not, I wish it was always beautiful like this. Like it's full of peaks, it's full of things happening. It's a lot of events, so you need to go and understand them. So, okay, when we do that, however, experiments, you can imagine, are much, much slower than the pooling in the computer. Because in the computer, we do that, you're simulating nanoseconds, right? You're simulating maybe microseconds. They are not that fast with an AFM. They cannot go up and down in nanoseconds. So they need to go up and down in a second. They need to go up and down in a millisecond. So how do we actually convert those things? There are a bunch of theories for that, but you're going to get something like this. Like you're going to get a curve that you can see your experimental data down here and our simulation data up here. So the way we do it, why we call this in silico single molecule force spectroscopy, we call it spectroscopy, is because each point that you're seeing here is a simulation. It's like a hundreds of nanosecond long simulation, sometimes microsecond long simulation, even more than that. And some of here are faster, like nanosecond simulation. So we actually get 2,000, 3,000 traces, 3,000 simulations of the same system to recover the kind of behavior that you have in the experiment. And you see this huge distribution of forces. So you're seeing force by force loading rate, that's the speed, basically. And you see that sometimes I get 5,000 piconewtons to rupture, or sometimes I get 3,000 for the same velocity, for the same pulling. Okay? So we are doing, and if you look at experimental data, by the way, it looks exactly the same. So we are doing what they do in the experiment. So we are trying to reproduce the experiment so we can actually predict. So in this case here, the experimental data is excluded from the fit. We're only fitting our data, and we're getting the same results from the experiment. So we can predict the experiments. Okay? Then in our lab, we do that. We do dozens of, maybe sometimes thousands of simulations. We do a lot of network analysis that Marcel is going to talk to you about, how to analyze that. And we use a lot of dimensionality reduction tools, try to use AI to predict stuff, and that's the kind of thing that we have been doing. Okay, so in our lab, although we are in physics, we love this equation, but we ignore it. We are gonna just do F equal ma, that's hard enough, and you're gonna keep doing kind of simple Newtonian. It's just so much easier, So you're gonna keep doing that. And so what we also, just to give you an idea, we develop a lot of things here, and we work on a lot of different mechanoactive proteins like bacterial adhesions, virus adhesion, like parasite adhesion, muscular proteins, drug delivery system. We work on a lot of different things that are mecanostable. Okay, so like it's not drug delivery like regular. We're interested when you need mechanostability. And we also help the development of we have been leading the development of the network analysis. Marcel and I led the development of QMM in the past. We're the team that develops NAMD, QuickMD, QuickFold, GMD2, all of that. So we have been working on a lot of these projects here. And although I'm not going to talk about that, I'm just going to show to you, just because we didn't talk about that at any moment, but you can also use VMD and NAMD to calculate UMMM stuff. You can use for quantum mechanics, and you can even see beautiful orbital images in VMD. You can track in a QMMM dynamics how the energy of homolumin orbitals move, and you can see how the energy is going up and down. You can do all of that with VMD and NAMD, just not the topic. Okay. So why do I care so much about mechanobiology? So mechanics is very important in proteins everywhere. So when I was doing my PhD, I was studying this guy, the K2PK potassium channel. We were studying basically the opening and closing mechanisms, the presence of anesthetics. So all of that happens because of a mechanical, it's not a mechanical active chain, there are many of them there are, but like this happens because of mechanical transformations in the membrane that pull these arms here at the bottom and make a rotation in the channel that opens the channel and closes the channel. Okay, so mechanics, protein mechanics there. This happens at very little force, by the way, okay, just a couple of piconewtons. Also, there are other things like filaments that I work together with Ulla Petikainen from Finland, and she was interested in these filaments, how some mutations in filaments, and I can show more details here, how mutations in filaments, both on this ROD2 region and this ROD1 region, were actually related to some genetic diseases that are quite nasty. And these are very simple mutations, like you have one or two mutations, and what happened is, because these filaments work as mechanical linkers, they release biochemical signals. The way they do that is they have inserted in between all these complexes a few peptides. You can see here one of them. So this orange part is a separate peptide that is just isolated. So what happened is you form the complex and the filaments are under stress. If you put too much force on them, if you put too much stress on them, they unfold a little bit, this peptide now is released. When the peptide is released, your cells know that there is force being applied, and that is going to initiate some other process inside your cell. The problem is, if you have a mutation, and it's down here, the mutation, like a tiny mutation down here, if you have a mutation there, the peptide that is in the other side, it's actually released earlier. So that means lower forces can activate that biochemical cascade that causes a genetic problem in your heart. So there are some other ones similar in the other side. So like if you unfold, if you break the complex earlier, you end up releasing some signal, and that causes. So there's a lot of important things related to this mechanics. And these are filaments inside your cells. They're sensitive to like just a couple of piconewtons. Like it's a very small amount of force that you need to make that happen. If we go to the other end, you probably heard about strep-avidin biotin. You know, like that's one of the most used things in biotechnology. These systems are like considered to be like the strongest things ever, you know, because they can form complexes that never rupture, and almost never. And so what they do is we were studying in the past that why some experiments, the question was, why some experiments show that they rupture at 200 piconewtons and some others show that they rupture at 400 piconewtons. To be honest, when you see the result from single molecule experiments, you would see a huge distribution coming from like 100 piconewtons all the way to 400. So why it ruptures at 100 and 400? That's a huge difference. And we actually figured that out. We worked with Gal in poolings from different points. And what happened is, just to explain quickly to you, when you do an experiment, you don't necessarily hold the system at a specific position. Most of the times when you're doing experiments with the systems, like in molecular biology, you use what we call reactive amines. So you're going to bind it to any amine that is available in the surface. So sometimes you might be doing your experiment by creating a linker here at the N-terminal as an amine, or you might be here in the middle because there is an available lysine. So or on the other side, or it can be anywhere. So because of that, the mechanics, and you can clearly imagine that, right? Like if I pull like this, or if I pull like this, okay, a phone is very strong, but like if you pull in different geometries, you're gonna get different results. There are easier ways of, the easiest one to imagine is this. You can imagine, do this with your hand, right? If I do this, very easy to bring in and out. If I do this, I'm locked. So depending on how you apply the force, it might be easy or might be hard. And that's what was happening. The experiments were not precise because you're not doing a precise anchoring. And that was confirmed by experiments because we could simply anchor on the C-terminal or the N-terminal and we would observe different results. But then we went ahead and also did some extra studies where we were anchoring many different points. And then we got very different, like, so what you're seeing here in these arrows are like, what is the direction of the force compared, like if you're anchoring one point, pulling from different ones, how is the direction of the force that you're applying to the system? And actually, depending on the force, the direction, you get very different results. And we could decompose that, understand what the force propagation pathway, understand how that happens. Pretty cool, right? You can understand a lot of the mechanics of the system. But everything that I'm showing to you something that we knew now since the 90s, that we have this, most of the proteins kind of weak, they behave, they can do their work with below 100 piconewtons, to put it that way. Almost everything we know is happened here. Unfolding proteins, molecular motors. It's just like between a couple of piconewtons, maybe even below that, up to like 100 piconewtons. 100 piconewtons is like a strong one already. And then you have a strep avidin biotin, which is way stronger, right? And you have covalent bonds. So you have biology and you have chemistry. You know, biology is here, chemistry is there. You have this huge difference. Here, by the way, is like 1,800 piconewtons is the weakest bond ever measured, like for a covalent bond, and that's a gold-thiol bond. You know, like that's the weakest one that they could measure. The limit to the other side is, of course, unknown, because to get there, you would have to have something stronger as a handle, and there is no such thing. So like, we don't know what is the strongest bond. Okay? Under force, I mean. And by the way, this range of force that you're seeing here is at experimental velocities, you know, because remember I showed you, depending on the velocity pool, you get different results. Then what happened is in 2014, we discovered this guy. So we discovered that some cellulosome proteins could actually be stronger, and that's all in collaboration with the Dobbs lab and doing experiments and simulations. We could explain the mechanism, and we also found a bunch of them later. So we found that a lot of these cellulosomal complexes are actually super strong, getting all the way to the nanonewton regime of force for rupture. And of course, understanding that was very interesting. So we worked with at Bayer, who discovered the cellulosomes in the 80s. And the cellulosomes are these complexes that are very efficient in degrading biomass. So they were very important for this biofuel industry, the second generation biofuel, and also for biorefineries. Like how can you use, how can you convert like plants into polymers, you know, into like plastic. You know. So the cellulosomes we're studying, so they form these non-covalent bonds, they form these huge structures, and they're formed by cohesin Dockery interactions. And the one that we were studying the first was this one here. And the reason why we thought it would be interesting to study it is because if you look at this drawing from before our paper, like from the biochemistry paper that was from Bayer's lab, they're attached to the bacteria, and this is their cellulose substrate, so like their food. And they have the CBM, their carbohydrate binding module, so they're going to attach to their food source, and they're going to have this non-covalent interaction holding them to their food source. So you have something that binds strongly to the surface of their food. They are large, and they have this non-covalent interaction holding the entire bacteria in front of this. Okay, the bacteria is pretty large compared to this, right? So it's holding the entire thing. We thought this should be made on the shape. And then we went to study it. And that's what we found, right? They're super strong. And it was super interesting. You know, basically what you're doing is this, you know, like you have bacteria, you have cellulose, you have this thing holding everything together. We did the FMD I showed to you, we did a bunch of simulations, and this is what we got first in 2014. And that became the strongest protein complex in the time. So these were the simulations, these were the experiments, and we could kind of predict each other. We did a few other with some other systems. We actually found that some other were even stronger. We found some that were bimodal, meaning like they could be stronger or weaker depending on the way we do it. But the mechanism was the part that was interesting, you know? The AFM could have given this information to us, right? We found a stronger one. But what made this work very interesting is because of this. The simulations were able to show to us that this is the system before we apply a high force. And this is the system after we apply a high force. What happened is you have those systems, like it's a true kind of flat surfaces, they're here. If you don't apply any force, they form and unform normally. They're not special. They have a regular KD. But if you start applying force, what happens is somehow the side chains of everything reorganize and their contact area goes up. So if I try to pull them apart, their contact area that we're measuring went up. And because of that, they became stronger. There's no more hydrogen bonds, no more, like there are no extra things happening other than the contact area. So they just start to have this larger interaction area, and because of that, they become stronger. And so this is the first time someone could show how a catch bond, the name of bonds that get stronger under force, we call catch bond, how a catch bond worked at the molecular level. Okay, so this was one of them. And to give you some perspective, what with the analogy we made is like a molecular finger trap, you know, because you can easily form an unform if you go very slow, or if you just let it there. But if you apply a strong force, they lock, you know, and they become very strong. Cool, right? So simulations could help us with that. So back to our thing here, we are here now in this level, but then, you know, we can make it better. We discovered that bacterial adhesions that are very common in hospital infections are actually stronger. They're in the same level of strength as a covalent bond without covalent bonds. And this is actually quite impressive. Our lab has worked on that system a lot recently. And this is kind of where biology starts to challenge chemistry. You know, like, is the poor man covalent bond? You know, can we do a covalent bond without a covalent bond? And that's basically what the bacteria evolved. And if you think about in evolution perspective, that's very smart. I know the bacteria is not thinking about that, but it's quite smart. Because if you want to make a covalent bond, you need to have an enzyme that comes and form that bond. And if you make a covalent bond and you want to break it, you need another enzyme that comes and break it. So it's actually quite expensive for the bacteria to produce those things. If they come up with a protein, but they need the original protein that's making the bond already as strong as a covalent bond, they don't need all these enzymes. You know, they can just do it immediately. Okay, so, and I just like to say that this is something that Klaus said in an interview that I love. You know, biology starts to challenge chemistry because we created those labels, right? Nature does not have labels, right? So there's no such thing as physics, biology, and chemistry if you're studying nature. Okay, so the way it works is, imagine you have a bacteria, the bacteria comes, binds to a surface, interact with the abiotic surface that can be like a hip implant. That's the most common way of getting these infections. When they're on top of that surface, they might feel some hydrodynamic drag, you know, like because, you know, blood moving, or when they infect urinary tract, you know, like you're peeing. That's actually the strongest force that we can feel. And then that we know, then you're going to have some adhesion force. You're going to try to hold each of it, right? And then if they do that, they're going to start forming biofilms. So we are interested in this part here, the initial part of the infection. So like how they adhere to us. They do that using this kind of famous system, the MM-SCRAMs, and these are these adhesive matrix molecules that bind to basically everything we have in our extracellular matrix. So they're going to bind to anything like fibrinogen, like fibronectin, keratin, collagen, elastin, basically anything. You know, like whatever you have outside of your cells, it can bind you. Okay? And depending on, so basically it's going to be depending on what you want to bind to, you're going to use a different one, but they all look the same. Okay, so, and this is for Staphylococcus epidermidis and Staphylococcus aureus, which are the two most common bugs for the interactions. So when you look at the SDR, just to give you some idea, we're working on a bunch of these systems now. This is somehow how it looks. So you have a host cell, so the human, you're going to have something that they bind to, like in this case here, you have adhering, and you're going to have the protein in the middle, and then you're going to have part of the protein, have a bunch of shock absorbers in the bacteria. So the bacteria part is the red, blue, and green. The orange part is human. Okay? They go bind. The way it works is this latch, this green part is open, like making the pocket available. When this finds the human protein, this latch comes on top and closes. And when it closes, it has all this chain here and kind of gets there strong. When you try to pull on it, these B domains, we showed that a few years ago, these B domains work as shock absorbers. So they are also super strong. They're actually the strongest folds we know. And they rupture a little bit before you would rupture the bond. So they're also nanometer strong. So, but before you rupture this guy, you rupture those. So allowing the system to kind of relax a little bit, try to survive more and keep strongly bound to you. So the way we did the experiment, we then holding this whole thing here, right? So this thing is holding you to basically holding to your cells, holding the bacteria to your cells under flow. So, and then we're gonna have did AFM with that, and also did the simulations. So Lucas Miles, who now just moved back to Germany, he was in Germany, moved to the States, now he's back in Germany, did the experiment, and he was in Herman's lab, and he got these amazing high forces, and I was there in Munich when we were doing together, and he came back from the lab. That's a funny story. He came back from the lab, and I broke my microscope, you know, because these results were insane. You know, that's what he thought. He broke the microscope because, you know, no one have ever seen forces like this before. And then we were doing simulation at the same time. I started downloading the results and like, oh, maybe I broke the supercomputer because we're getting the same thing. And then that's how we started working on that, trying to understand that system. And then we got that curve that you seen before, you know, like this is from the paper. This fits this model here. There's a Duduku-Hammer-Szabo model that shows that the force should increase with the loading rate. But if you look at this, there was still 5 orders of magnitude of difference in pulling speeds. And we have been working on that. So Diego worked with that before starting digging the VMD, a lot, like improving VMD a lot. He worked on that, trying to meet the loading rates of the experiments with the loading rates of the simulation. So we're using like some coarse graining. And we have been doing that. We have been successful at like completing the two work. Then I'm going to explain to you a little bit what is the mechanism of the system, which is pretty cool. But before that, you might be asking yourself, why are you doing 2,000 simulations? Like, why do you need so many? I can give you this thing, one example. This is from a test that we did. Just look at the wild type system and a mutant. You're interested in seeing if mutating something in this system is going to make it weaker or stronger. And then you can go there, measure once, and you see that this force is the system. And then you're looking at the system like, oh yeah, the wild type is stronger, it's 4,000. The other one is 3,600. And most of these simulations are done like that. You do it once, and then you got the result. And like, okay, the one's weaker. But then your experimentalist friend come and tell you, no, that's the opposite. And if you start running multiple simulations, you're going to observe that it's actually different. So be careful. Simulations are just like experiments. You know, like you have to run many of them to get a correct distribution of the system. And the only thing different between these systems is that your initial velocity. The system is exactly the same. It's just like it's a stochastic process. It can happen different forces. So if you want to start getting some nice results here, our experience is that if you not plot this histogram well, you're going to lead 20, 30, 40 replicas. Otherwise, you don't get. And you might need more for some systems. Okay? So be aware of that. You know, that's something that you might need. And that's the thing that you're saying. The higher impact journey you're going into publish something, you're going to see more and more replicas like this. You know, like if the MD is important, like large part of the publication. You might be doing just a small contribution, then you don't see so much. But if it's a large portion, that's what you're going to see. Okay, what is the mechanism behind? Because I want to give the time to Marcelo. Initially, when that system, the Hisian system was published, the crystal structure was published before us. They did not know about the force, but they knew like, okay, this thing should be strong. And they solved that they had these two phenylalanines blocking the channel. How you have the latch, you have the two phenylalanines, and then they call it in the paper the bougie plug, because they thought like, if you have something large, you have a tiny constriction, fast through it's going to be.
I'm having trouble getting OFI to build, and it might just be I've got the wrong modules selected. Can you provide details of how you actually got that stuff? Yeah, it's not.
So they just told me that I'm not sharing my screen.
Okay. Yeah.
I was sharing before we start changing the thing.
You just put them in Slack.
No problem. Sorry about that. Okay, so
Kirk, I think you're unmuted.
we have this. Okay, they were talking about these bougie plugs, you know, so they're talking about these large amino acids being responsible for this being strong. So you have something large passing through a tiny constriction that should make this strong. So then we went ahead and tested that. You know, let's call the wild type has two phenylalanines, so that's what's called at 100%. In vitro, in silico here, so that's the force they measure in the experiment. That's the force we measure in the computer, because remember, we pull faster. Then we put an extra phenylalanine, a third one. That's because the crystal was actually solved with 3 because it was easier to crystallize. So then we did the same thing. Goes a little bit up, goes a little bit up. Okay, good. That's a good sign, right? So you're getting more stuff. It's going to be stronger. So maybe if we had 10 of them, it's going to be super strong. But let's go the other way. Let's try to take them out. So then we removed. remove to just one. Well, still kind of the same. And then remove another one, more or less the same, right? Still strong. So without the phenylalanine, was still super strong. So that's not the reason. When we start looking at the simulations more and more, so basically the AFM work, the experimental work would be that here, you know, like, because you will have to come up with another solution, which is not gonna be easy to understand the system. Then we start looking at the hydrogen bonding, and we observe that there's a bunch of backbone interactions. Okay, beta sheets, right? No surprise. A bunch of backbone interactions, and you're mapping all of them here and the strength of all of them. And we notice that they form this twisted shape. And that started giving us some hint like, okay, you have a bunch of hydrogen bonds at once, they break all at once, and you have this twist. Okay, why is the twist important? Because these are flexible linkers at both ends. And this thing has a perfect shear geometry where it's going to be strong. Otherwise, it's not strong. You're going to explain why. So now, if you have flexible linker on both sides, it doesn't matter if you pull like this, or if you pull like this, or if you pull like this, it's always going to align, and the system is going to be in a line. Having bonds pointing in all directions, make sure that you're never peeling the bond. Because it's very cheap to peel bonds. Like titanium, you peel. That's why titanium is not that strong. Although it's strong, it's not that strong. Because you break one hydrogen bond at a time. In this case, you cannot do that. Because you have a bunch of hydrogen bonds in every direction. So you go back to this aligned position. And when you are aligned, you form all the hydrogen bonds again. And you always have like a bunch of hydrogen bonds to break at once. More or less, we could count about 17 hydrogen bonds that you have to break at the same time. So if that's correct, if we are correct, if that's the mechanism, that means that you don't need side chains. You only need backbone directions. And that would make this super strong. And of course, we have computers, we can play God. And that's exactly what we did. Let's change everybody to a glycine. And in the computer, we change every single amino acid to a glycine. And boom, you're still in the nanonewton regime. Right? Okay, it's not the same force because there's some confinement there that makes the final adjustment. But bear with me, like we are still in the 2.5 tube nanonewton regime. That is way stronger than any other, like it's an order of magnitude stronger than strep-avidin biotin. Okay, so this is super strong just with the glycine. But you can tell me, yeah, that's crazy stuff that you did in the computer. You know? But we were lucky because there is another adhesin that binds this K10 peptide that is basically a bunch of glycines, only a few serines in the middle. So it's like a very little side chains. And then we measured that both experimentally in the computer, and we could see there was also in the nanonewton regime, and they were actually super strong as well. So the same, but the same. So at the end of the day, what we could show is that this system was strong because you have to break a bunch of hydrogens at once, and those are backbone interaction, which is also very interesting because that means that if this protein binds to something new, let's say if there's a mutation that makes it bind to a new peptide in your body, that's going to be strong. It doesn't matter what it binds, it's strong. So the side chains are very important for affinity, okay? Don't get me wrong. Like you're going to bind only if your side chains, you have a distant affinity to what you're binding to. But if you bind, it doesn't matter the sequence, you're going to be strong. Because if you're bound, you're going to get into this perfect shape geometry that is going to make it super strong. Then the question at the time was this really a catch bond? We could not measure that because I mentioned it briefly before. A catch bond is a bond that as you apply force, it lives longer. So the more force you apply, see the bond actually lives longer until a certain point. You know, there's a limit, of course. So a slip bond is a common bond is the opposite. If you apply force to a slip bond, it lives less. So like if you apply tension, the bond is going to rupture earlier, which makes more sense, right? But the catch bond is the opposite. The more force you apply, the longer the bond leaves. And remember this case. So the problem is when we did the experiments, we could not do what they call a force clamp experiment. That is the AFM holding the system until the rupture, until the bond breaks. But 2 years ago, a group in Singapore was able to do that with magnetic tweezers. Yeah, sure. Sure.
I mean, I just want.
Oh, good. So this group in Singapore was able to do the magnetic tweezers. And the reason we could not do that in the AFM is because it doesn't matter how good your AFM is. And Galb's lab produced their own AFM. They're amazing. You have drift, right? Like, so you go up and down, but there is some drift, lateral drift. Especially if you take minutes measuring the same bond, because you see you're going to start moving. You know, during the time of going up and down, the drift is zero. But if you keep holding for minutes, hours, that you start drifting, and so you cannot measure. With magnetic tweezers, more stable. They were able to do it, and they got to points where this thing could resist up to 7 hours. So they could have a bond under force holding for 7 hours. The maximum they could get. On average, they were getting 2 to 3 hours, if I'm not wrong. To get give you an idea, these bacteria have the mean time for them to replicate is 50 minutes. So they can actually grow a culture under force, basically. You know, like it's like this. The system is actually super stable under force. And then we had a lot of simulations like that. So we decided to test and check. So that is a catch bond, answering your question, right? So, but we actually could explain the mechanism. We saw that if you just hold the two ends, like the magnetic tweezer experiment, the system was still kind of flexible. If you apply higher force, like the AFM experiment, the system was super stable. Marcel is going to talk more about that, but like he was able to measure that. So what you're seeing here is blue means a stronger correlation. So meaning he was able to see that the more force you apply, the more stable, more rigid the system gets, and the more like the system becomes stable as a whole. And so blue means stronger in the FM, red means stronger in the clamping, like magnetic tweezer. And that was showing like how the system is actually getting more rigid as you apply force, and that's why it's super strong. Also, like as the catch bond mechanism behind. At the end, you can imagine something simple like this, and Chris showed something like that too. If you want to go from A to B, meaning bound, unbound state, you can follow a very easy path, right? That's the solution path. You're going to just follow the valley here. But if you apply force, like a crazy high force, you're going to try to go straight. You know, that's you doing the FM super fast, or us super fast with the simulation. Of course, you don't go exactly straight. You find some other paths. And so depending on the force you apply, you kind of block the road to go around and only find these other paths. And so you're basically reducing the energy barrier to go through the mountain instead of going through the valley. And that's kind of what you see in this system. So you block one pathway and find another one. So you can actually have different pathways, a dissociation and association pathways, and that's what we're seeing. That's the catch bond part of the system. Should finish because I passed my time already. How this system became so force resilient. That's something that Priscilla in our lab is studying. She discovered something that is probably, so if you found that, because at least I find cool, that we discovered that this system is super strong in how that works, she found something even more cool because she was able to see that it looks like they're evolving to become stronger. And she noticed that because we were testing basically all adhesins with AlphaFold and then doing some simulations. So we were doing like 20 replicas of pooling for each one of these adhesins. And then she noticed that the ones that were isolated more recently, the ones that were methicillin resistant, were actually stronger than the ones that were methicillin susceptible. And that was like a dirty signal because she was doing like very quick and dirty simulations, you know. But that's like, oh, interesting. And then we start studying that. Okay, let's all be careful with that. Can we see that really? And for instance, for one of the mutations, one of the points, she noticed that one mutation, this is one single mutation. So this is the force that you get. And she was trying to get every single amino acid in that position. It's position 322. And see, this is the methicillin resistant one. This is the methicillin susceptible one. So this is the one that is antibiotic resistant. This is the one that can be killed by the antibiotic. So if you think about the evolution, this is probably older than this one. And you see that they are stronger than the one that is methicillin susceptible. And that's actually the stronger, is the amino acid in that position that make the system more mechanostable, more mechanoresilient, right? Since then, she has been doing a bunch of experiments, sequencing every single one of these adhesins that we can get our hands into. So she moved from the computer to the bench. She's using the lab of a colleague here in the college, like he's in biology. And she is, we got our hands into some historical strains. That was the coolest thing. I don't know how they did that, but we're very happy that someone decided to store bacteria from the 1800s. So we have those bacteria, and then we actually got the genome of those bacteria. They were not sequenced before, and with all of that, we actually are now studying how they are evolving for us. We have some preliminary data on that. I don't have it here, but it seems to be that they were much weaker than the ones we have today. So it looks like we are actually helping these bacteria to become stronger and stronger, and things are getting worse and worse, right? So the take-home message to you is, so there's this thing of like going from bound to unbound state, you can follow different paths, right? So we can use these systems as if it was an experiment. And I hope you understand that, you know, like we don't run simulations that just put the system there, let it run and see what happened at the end. We do experiments with it, you know, like we go there and like, what if we change this? What if we change that? That's the smart way of doing MD, okay? It's like doing an experiment. You don't just go there and observe. Yeah, you can, but that's like, it's not a science paper anymore, right? So you have to go and change things. You have to go and understand this. You have to go and test hypotheses. And that's the kind of thing you can do with simulations. And my other message, because of all these things with this adhesions, is may the force be with you, right? And by the way, I just showed this because it's cool. So this is actually done with VMD. So you can export your VMD to your PowerPoint and it become a 3D object that you can rotate inside PowerPoint, by the way. So this is something that probably most of you don't know. And just want to say thank you to all the collaborators, the people that helped this, the funding. And if you have any questions, I'll be happy to answer. I just want to show this to you again. So see, this is PowerPoint. So this is how my group now shares stuff with the experimentalists, the collaborators, because they are not so familiar with using any of these tools like PyMol, VMD, anything. So we just share with them PowerPoint, and then they can go and check, they can zoom in, they can, oh, interesting, like we can, there's another button for some that don't remember where it is, but like you can go and check, and then you can make those animations like the one I made when you, well, PowerPoint can handle those. Yeah, thank you. Any questions? Yeah.
So I'm just thinking about the last example, the antimicrobial resistance. Is there any, other than developing resistance to the drug, is there any advantage to being like stronger? Or like if you remove the pressure of the drug, would it not be selected for?
I don't know the answer. So what we think is the following. So first of all, the no antibiotics work on adhesions. Okay, they work on something else. The reason why I think adhesions are important on this, like adhesions are evolving to become stronger, is like our initial guess is the bacteria that were selected because they can stay strong, like bound to you, they have a much smaller chance of being attacked by the antibiotics, because if they have this biofilm, it's actually much harder for the antibiotics to work on them and all that. So if you can keep them strongly bound, they probably have a larger chance of having other mutations that, like, because they just like survive longer. If you survive longer, there's another chance of another mutation that can keep you there. So like basically it's just selecting for the ones that can be more stable. There's another part of it that is interesting, is that in 2005, I think, it went for clinical trials, a drug for adhesions, as a vaccine actually. And they were like 100% sure it would work, because in vitro was showing that they like would bind to the adhesions. However, they were binding it to the wrong side, because they did not know the mechanostability mechanism. They would, if they knew what we know now, they would be like, okay, that's not going to work. They went all the way to clinical trials before they failed. So this shows like how the simulation, how understanding the system is very important for developing drugs, although we don't develop drugs in our lab. So, yeah.
Like, how do you measure or identify the nitrogen bonds? Which tool do you use? How do you use it?
So we use VMD itself. There's a hydrogen bond tool there. In molecular dynamics, we don't have hydrogen bond, right? So we don't have those. So the way you detect them is by geometry. So you say that if you have a bond that is kind of aligned up to like a 30-degree angle at a certain distance, that could be formed. If it was a quantum mechanical calculation, that would be a hydrogen bond. But we don't have a hydrogen bond per se. So, but like we know that that position would form a hydrogen bond. The distance is, I think, I don't remember exactly, it's 3.5 angstroms between the donor and receptor. It's the distance and the angle between the bonds. Because, you know, like if you're pointing the other way, even though it's close, it's not a hydrogen bond, right? So, yeah. Yeah. Like something like this. Oh, no, this is just a drawing that we made for explaining the system. To calculate this, it would be actually quite complicated. That's the kind of thing that Krishipo was talking the other day. But like, if you want to get a complete map that shows multiple paths and get the whole picture, it's a lot more calculation than we already did. So but it's doable. It's just very complicated.
What language technique do you use?
I would probably use string on this one, make a string for each one of the paths and calculate them both. But maybe Chris can have a different opinion. Like if you have something that unbinds through different paths, a mechanical and a mechanical one, what method could?
Calculation would give you one, but there would be many possibilities. The graph also here you have a bifurcation. So
He would have to calculate both paths.
you can just cluster. There is a possibility. Or it's temporal.
Yeah, this is this is definitely complicated. Yeah. Let's put it this way, even giving this as a thesis for a good student would be hard. Probably it's going to change. I don't think you can jump the high force one here for the adhesion because of changing temperature. To be honest, I think if I increase the temperature, I go for the low force always. Because you would end up breaking the hydrogen bonds early, and then you would open the At the end, what this means is that adhesion system is here, you have to break all the 17 hydrogen bonds at once going like this. The low force pass is the one the magnetic user can also see, is when you open the latch and the peptide gets out. So you don't break all the bonds at once, you peel it off, you peel it twice, you peel the latch and peel the peptide. So this is kind of what we have as a observation. So if you increase temperature, there's a much higher chance you're going to go through there.
Is there a barrier to go from the bound and the high force way before it gets unbound?
Yeah, it's a huge barrier.
No, I mean, that's unbound. Like just to choose the two pathways, is there a barrier? Or can you just diffuse into the
No, you cannot diffuse and get the high force one. I don't think so. If that's what you're asking.
no, is there a barrier to before dissociating? Is there a barrier to select the path, like a smaller barrier?
I don't get your question, sorry.
If
You're here, you're bound, you're gonna unbind. You're either gonna go one way where you're gonna open the latch and dissociate, or you're gonna go through a much higher barrier.
so let me rephrase, is there a barrier to form the hydrogen bonds that are
Form?
yeah, or are they always formed?
No, they're formed if you're bound. You get it? So that's the binding part.
can ask.
Sure, I'll let Marcelo continue, and Marcel, don't forget to share your slides, that was not smart for me.
Share my slides. No, of course not. We can leave